It is a great pleasure to introduce Alexander Lachmann. Alex joined the Mayan lab in 2009 when the lab was just established, and he joined as data scientist after he received his master's degree in computer science from Aachen in Germany. And he was extremely productive. He did really well in the lab back then. He was the lead author of two apps that really set the stage for a lot of the applications that we've done, Kia and Chia. And he was really demonstrating like excellent programming skills and a lot of creativity. Somewhere up in the 2012, he decided to do a PhD, and he joined the Califano lab where he at Columbia University, and he got his PhD. And then after he graduated, I convinced him to come back to the lab, and he's been here since. And his major project was this ARCHS4 project that you're going to hear about today. And ARCHS4 is not just having all of the geo datasets uniformly aligned. It's actually what you can do with the data. Everything that Alex did beyond developing that workflow that is amazingly cost effective is based on the ability to have all of this data available, and it facilitates a lot of other tools and studies and discoveries. And yeah, I think you're going to enjoy this introduction that Alex will give today to a completely new website that was developed, that he developed for hosting the ARCHS4 resources. And the other thing that is really happening with the ARCHS4 data is the ability to use those embeddings of genes in the space of all of those thousands and thousands of studies. And this really sets apart this data set in biology for gene discovery and gene function discovery and many other applications of AI, and some very exciting developments in this field that this resource is enabling. So Alex, take it away.
Yeah, thank you, Avi, for the introduction. Just to mention, I think you can add questions down below. I think there should be an option for you if you have questions. I think you can post them there. And so then, yeah, we'll start. So today I'm going to talk about ARCHS4. And so that was a project that started around 2018. And at the time we set off to align all of the RNA-seq data that was out there. And so this is now the update. This is like the first major update of all of our like pipelines and websites and tools that interact with this data. So gene expression data is really useful, and the current state of our understanding of this landscape is somewhat similar as like a medieval map. So this is maybe a map from the 1400s or so, and like while the outlines of these landscapes are not ideal, they are still kind of a useful tool to navigate and really, you know, are helpful in finding new discoveries in medicine and biology. So ARCHS4 is really trying to build this gene expression landscape, which is, you know, at this point maybe not perfect, but over the years and the data grows, these maps will be more detailed and more powerful to do tools. So ARCHS4 is an open access platform, and it has basically two components. It has a backend that processes all of the available RNA-seq data in a very cost-efficient way. And then it makes those accessible to researchers through different means. And so the main components of ARCHS4 are human data and mouse samples. And then we have some other samples from some other species, but it's a smaller part of it. So yeah, what is ARCHS4, right? So as I mentioned before, the thing in the background that users probably don't interact with as much is the pipeline. And so this was completely revamped from the old version. So now this runs completely automated, and it basically discovers new samples automatically, schedules them for alignment, and then automatically packages the data and then like in these H5 files, and then makes them available for download. And then another big component is the front end, which we completely revamped. So this is hosted at ARCHS4.org right now. And so this is completely rewritten. Initially was like a PHP site. Now it's a React page. So there's not a single line of code that we could really reuse. But there is a lot of functionality. So you can have a data view where you can analyze samples or genes, and we'll show that in more detail. And then gene pages, which are really focusing on individual genes. And they add a lot of information, that might be relevant, like tissue expression, gene correlations, and so on. And then we also added additional functions to download the data in bulk, or like if you only want to download individual series, that is now also an option. And then a lot of the functionality that allows users to interact with the data in real time are hosted as a API. So these are open for users to access. So you can, find k-nearest neighbor signature searches, or you can identify marker genes through an API. And we also leverage a tool that we developed called PrismX that does gene annotations. So this is especially interesting for genes that have very little annotation. So there's a lot of understudy genes, and you can get a really quick idea of what these genes might be involved in with these machine learning predictions. And then lastly, which is, I think, also a very important tool, is ARCHS4py, which is a Python package that allows users to interact with the large bulk files that we offer for download. And these files are very large, they're about 50 gigabytes, but they contain all of the gene expression, all the metadata that the ARCHS4 processed. So if you're like a computational person, then this is probably the way to go. And there will also be an R package that has very similar functionality to this. So just to put things in perspective, when we started in 2018, there was already a lot of RNA-seq data out there. There was about 100,000 mouse samples and 85,000 human samples. And back then it seemed like a big task to process. So that's why we ended up using cloud computing. But as with all data technologies, it's kind of an exponential field. So just in 6 years, we kind of have an order of magnitude more data to deal with. So it's really essential that we're working with scalable systems that we can carry on in this like exponential regime. And so this is especially true if we think about how this data might actually grow in the future. So we obviously don't know if it flattens out eventually, but especially with the like more prolific use of single cell or spatial transcriptomics, this is probably still growing significantly over time. So just as a quick overview of the statistics, we now scratched the 1 million mark samples for mouse and also for humans. So it's a pretty rich data set. And I think one of the nice things about this data set is that it is a homogeneously processed set of data, but it comes from a very heterogeneous background. So for example, in mouse, we have 1 million samples, but they come from like 32,000 different studies. And so you can find, let's say, samples of interest that come from many different experiments, and you get a much better idea of the possible variation that you can encounter, rather than having like a very homogeneously processed data set like GTEx, for example. And as I mentioned, we also have this ARCHS4 zoo subcategory. So there's a couple of species, but it's only a few samples compared to the mouse and human. And as you can see over time, the number of samples grows pretty quickly. It looks like it flattens off, but this is just because samples only become available after a certain time. So as I mentioned before, so the challenges are really in the data processing. It's really like a networking problem because we're working with petabytes of data. So this is comparable to the data throughput of Netflix or something. If we would really, process this really quick, but it's a lot of data. And then the problem of sequence alignment is that it requires a lot of memory, a lot of compute, and you have to deal with flexible storage. And so designing the pipeline, we really had to focus on those components, and we kind of got it right at the beginning in 2018 with cloud infrastructure. But what we really struggled with, and which is maybe the harder problem, is how do you make that data accessible for people? So we, acknowledged that there's different users that have different backgrounds and different demands. So we really had to focus on both the graphical user interfaces and then also command line users or programmers that, maybe want to train a neural network and they just need quick access to the data. And yeah, so I'll also show some, if we have some time, like a quick demo of this Python package. And so as I mentioned before, all these challenges need to scale with exponentially growing data volumes. So this is tricky, especially in the front-end development where a user expects real-time results. And so that's something we'll touch on. So the pipeline in its general idea hasn't really changed much. It just evolved into like a much more robust system over time. But at the moment, we have a Dockerized workflow, and we have a job queue and the server that hosts these jobs. And once we detect that there are samples waiting in the queue, the cloud spins up, let's say, 500 computers, and then processes all the RNA-seq and stores all the data in the cloud. And then there is processing after that data is aligned. And so the key is really to keep the cost down, because if you have to process 1 million samples and you have to pay 10 cents per sample, you have a problem, right? So at the moment, we're way below half a cent per sample. So this makes this feasible, and also feasible to maintain this for like a long period of time. And so this is kind of how the processing schedule looks like at the moment. So we do sample discovery once a week, where a server goes out and searches for new samples and then puts them into the waiting, like into the job queue. And then the pipeline scaling is dynamic. It will check the queue all the time. And if it sees new samples, it scales up and processes them right away. And then we package the data once a month, and then upload them under the latest tag. So if you want to stay up to date, like within a month, there might only be 20 or 30,000 new samples, but it could also be more. And then, so once these are pushed, we also update automatically all the data APIs that serve or interact with this data for the graphical user interfaces. Okay, so the new website, and I hope the website survives this seminar. And like, if there's a lot of people trying a lot of things, it might struggle. So I hope this works. But we have a new landing page where we have now like a news section, which is new. And we also have a nice overview of where the data is coming from all over the world. So it's really like a global resource of gene expression. So it's basically coming from every continent at this point, which is really nice. And then we also have this pipeline activity monitor, which is especially nice for me because I can just look at the website and I can see the status of the pipeline, if it's processing at the moment, when it looked, for the last samples and so on. But you can look at it too. It's a nice feature. So then in the menu, you have a couple of points. I think for most users, the Data Explorer is the most interesting one, and that's the section that allows you to really dive into the gene expression, do some queries on it, and then extract submatrices from it on the fly. So this is what it looks like, and if you know the old website, you can see that it looks very similar to the old version, and this is by choice. I wanted it to not look completely different, but it should be more performant and add additional features. So the Explorer has two views. It has a sample-centric view, and it has a gene-centric view, and you can toggle that in that menu between sample and gene, and you can toggle between human and mouse. And the first thing you will see is this UMap of the three-dimensional representation of the gene expression. So each plot is about 1 million data points. And so you really get an idea and appreciation of the data at this point. There's a lot of discussion if UMaps are useful or misleading, but I think in this case, it's just, like a good inspirational tool to really see, how things, stick together and, yeah. And so what you can do here in the sample-centric view is you can search for tissue types, and then they will be highlighted in the plot. And this is in this result table you see on the bottom. And so now you can download, if you click on one of these download links, it will download, after like a little bit of loading, it will download a zipped file with the gene expression in it. So that was previously not possible. And then also new is, or like improved, is this k-nearest neighbor search. And so we have two options. You can either search with upregulated genes and downregulated genes. And it will then try to find gene expression profiles where the genes are overexpressed or underexpressed relative to the other samples in ARCHS4. And it can do this more or less in real time. And then we also have a weighted profile search. So there you would just give it a full gene expression profile with the gene counts. And then it can identify samples that have a similar structure. And so this was actually quite a challenge to get this to work fast. And so here's a quick overview what it does. So we use something called the Johnson-Lindenstrauss transformation. And it basically is a dimensionality reduction technique. We take a high dimensional data point and bring it into a lower dimensional space. But the key element here is that you keep all the pairwise distances proportional to each other. So if you have three genes, not genes, like three samples, and you have sample A is more similar to sample B than to sample C, then that is kind of preserved in this projection. And so in the figure, you can see that we kind of pre-process all the dimensionality reductions for our database. And then if a user comes with a query, we can project that in real time. And then we use a cosine similarity search to find similar profiles. Okay, and so at the moment, we're using 200 dimensions in the projection, and we can kind of show that we get like a fairly reasonable similarity, like a similar result as if we would use the full dimensionality, but at orders of magnitude faster query time. And so then this is the sample-centric view. Then the next thing is the gene-centric view. And what you can do now, because we have this capability of querying the data in real time on the back end, users can search for any term that they're interested in, but like a tissue type or a cell line or something like that. And so this search function, the marker gene function, will identify genes that are upregulated relative to a random background in ARCHS4. And that also can run in real time. So if you submit something like this, and you can choose how many genes you want to get returned. You get these in the result table. And as a nice feature, you can also then immediately submit them to Enricher. So there's a small button. And then if you click that, the genes will be submitted. And you can look at the enrichment results. Okay, so then we have another like very important part of this tool, and that's the gene pages. So the gene pages, we had them already in the old website, and they had like a brief description of the gene. And then we also had some functional predictions where we used the gene expression and the correlations between genes to associate genes to other genes and then transfer their functions. And so this was a really nice tool that we really tried to enhance on. And so the gene pages now, they have similar functionality, but we improved them like across the board. So the individual components of this are so for one are the gene descriptions. So before we used descriptions from NCBI, and they were sometimes quite old, like from 2014, and they don't really get updated much. So we developed an AI-driven, an AI-powered system that can read publications, and it can cross-reference them with databases that maintain gene to publication relations. So we know if a gene is cited in a publication. And so these are then produced by aggregating up to 50 publications and then summarizing their results. And so for the first run so far, we processed about 500,000 publications and built a partial set of descriptions. So we still have to run a second, like a second group of it. But I think so far we have about 12,000 enhanced descriptions. And so the nice thing with this is also that the AI tries to find different contexts for the gene. So it's trying to be like, if there's multiple functions, it tries to capture them. And then if it finds relevant publications, it will also cite them in the abstract. So there's these embedded URLs, and you can click on them and it will bring you to PubMed. So you can read more about it. So I think this is like a really nice new feature. And in the case that the gene doesn't exist, then there's a fallback. So then you just get the small description. But we'll work on it to get it to work for every gene in the future. And so for this, we really use state-of-the-art AI models. So in this case, we're using the OpenAI O3 language model to do this, because I think, yeah, for this, I think it makes sense to just aim for the highest quality as possible. So I hope these are useful. And so another nice feature of the gene pages, which is like we got like requests for this a lot. And so this is the ability to identify genes that are positively and negatively correlated to the gene. And we had this in the old website where it would just show you a list of the most positively correlated genes. But it was pre-computed on like all of the ARCHS4 samples. And so the new feature now allows you to do this in a context-specific manner. So you can give it some meta information, so let's say lung, and the system will extract lung samples on the back end and then compute the correlations on the fly and then present them to the user. So you can potentially find very interesting differences in these contexts. And then this is something that hasn't changed much to the old version, but I think it's a nice feature. And this is the Gene Expression Atlas. And so this atlas tries to find the expression distribution in tissues. And then we have another one for cell lines. And we'll keep on improving this by finding, better definitions or like outlines for all the tissues that are out there. But so this is kind of the beauty of ARCHS4, that you get these things basically for free, right? Because we have all this gene expression, we can just build these atlases more or less on the fly. And so this is another case where you can really see that the diversity of where the samples come from really make a difference, because you really see that the genes show like a big distribution, so they're not, very uniformly distributed even in the same tissue type, because they come from so many different experiments. And yeah. And then so this is the gene annotation section of the gene pages. And so here we did a big upgrade where we went from the initial algorithm to annotate genes to an algorithm called PrismExp. And so this one is also using only high throughput gene expression data. So it's unbiased towards known biology, right? So if you have a gene with a lot of annotations, it doesn't really affect the algorithm prediction. And so it can predict the gene function. To briefly explain it, by splitting all of the gene expression in ARCHS4 into clusters, so it does an unsupervised clustering, and then it extracts machine learning features from all of these individual clusters, and then makes predictions based on those features. And so we have shown that this annotation is superior to what we had before. So now we have all these gene annotations for all these categories that you see on the left. So we have potential upstream transcription factors, gene ontology, pathway membership, disease associations, potential upstream kinases. So it's a pretty rich selection. And then you can also, so this is only for these sections. And then if you want to have more information, there's a link to the actual tool. So there's another website for PrismExp. And there you can also choose from all of the gene set libraries that are supported by Enricher. So instead of just having these 8 or 9 groups here, or like gene set libraries here, you can do that on all 170 gene set libraries that are in Enricher. And then now we get to the download section, which there we added this completely new backend. And it's a little bit invisible for the user, but so this is a pretty powerful data platform that we developed. And right now you have to sign in, but it's just using OAuth. We're not like saving any information from you. And when you log in, you can download the zip files that are specific for a gene set, for like a gene series, like a series, like a geo series. And in the zip file, there's a gene count matrix, and then there's also all the metadata. And so these are much smaller than just getting the big bulk data. So this might be more useful if you're just interested in a single series. Then you don't have to download like 50 gigabytes of data. And so this also this backend might be further used to add, like a very like advanced way of maybe potentially users being able to upload their own data and then processing it with the ARCHS4 pipeline. So that's something for the future. And then the main download section is somewhat similar to the old one. But so the big difference now is that the files get automatically updated once a month, and they get published as gene level expression, or they get or like also as transcript level expression, and they're always under the tag latest. And if this gets overwritten like once a month, and then once in a while we'll have a new version. And when that happens, then we have that below there as a separate. You can choose the version, so then you can fall back to like a stable version. So if you are worried that these files change, then just download the like a numbered version rather than the latest, and then that should be available for a long time. Okay, so now we kind of move to this programmatic part of the ARCHS4 project. And so the main thing that we use to distribute the ARCHS4 data is this H5 file format. And so it's a binary file, but it has really nice features in that it is like a hierarchical format, and you can store large data sets very efficiently. So it's like compressed. And the key thing is that it allows you to extract only parts of the data that you're interested in, which is really important for this gene expression matrix. So if we look at the fields, here we have this data folder, and then in there we have this expression object. And you can see that the dimensionality is 53,000 by like over a million, meaning there's 53,000 genes and over a million samples. And so either you can use existing libraries that support H5 and do it manually, or you can use the ARCHS4 Python package that we'll show in a little bit. And then we have the metadata, and this is kind of split into 3 groups. One is the meta information for the genes or transcripts. And so there's 3 fields. So it's the biotype. Is it a coding gene, or is it like a non-coding gene? And then we have the ensemble gene ID and the official gene symbol ID. And just to note, this is something people might encounter. The symbol might not necessarily be unique. So there is some duplicates because the ensemble gene level is the unique level. And ARCHS4py also has options to deduplicate those symbols if needed. And then for the metadata, the other big one is the annotations from the samples. And so this is extracting all the metadata from the Gene Expression Omnibus for the samples. And it's a little bit messy because there isn't very strict metadata enforcements in Geo. So that makes it a little bit difficult to navigate the gene expression using this metadata. But so these are all the fields that are supported here. And to highlight one of them specifically is the single cell probability. This is something that we compute. And it should be 1 or like larger than 0.5 if we think it is from a single cell experiment. And it's below 0.5 if we think it's a bulk sample. So if you want to filter out single cell and only want bulk data, you can use this field to do that. And then we also add the aligned reads, so you can see how many reads were actually aligned for that sample, which would be the sum of all the counts from that sample. Okay. So as a brief overlook, we have this ARCHS4 package. And as I mentioned before, there will also be an ARCHS4 package. And I'm just finalizing some more things, but it will have very similar functionality to the Python package, and it will have very similar syntax. And it allows you to extract gene expression. It allows you to do the metadata search. And the Python package can also execute the ARCHS4 pipeline locally. So I think this is very nice for users that have their own data, but they want to kind of put it into the same space as all the ARCHS4 data. Then you can just use this tool to do that. And I can go over that in a little bit. It's like very easy to use. And then there's some utility functions. So I can just demonstrate that real quick. So hope everybody can see this. So here, if you want to use it, you can just import this package when you install it, and then you have your path to your H5 file. Right? So if I run this, so one function, you can list all the fields. So this is something that we've shown before, but you can just look at what all the names are in here, which is useful later if you want to do some queries and you have to specify the metadata fields. And now there is basically 3 modules in this package. And one is the meta module. And as you might guess, it's there to allow you to query and search and extract metadata from the H5 file. So in this example here, we download the metadata from all the samples that match this search term. So ideally, it finds samples that are from myoblasts. And then the next line, so I'm running this right now. And then the next one will extract the metadata for these 3 geo samples. And then the other one, you can just extract all the metadata from all the samples in a given GSE, like a series. And then here you can also extract information about an individual field. So if I do this, the geo session, I get all the sample IDs. So in this case, because we have 960,000 samples, this vector will be 960,000 geo sessions. And then here I can just get all the gene symbols. And so to mention, you really only have to specify the last field name, and you don't have to specify the folder in which they're in. So this package just figures that out. Right, so if I show the sample metadata, so this was the metadata from these 3 geo samples. They're like in each row, you can get all the fields, and in here you can also specify what metadata fields you want to return. But in this case, we return the characteristics, the extraction protocol, and the source. Okay. And then the data module. So this is, I think, really interesting if you are, using gene expression programmatically for your workflows. And so there's a lot of ways you can extract data. So the first one is you can just extract a random set of samples. So this is useful if you want to build like a null model and you just need like a random background. And we have this flag here that removes the single cell data. So it's using the single cell probability that we talked about earlier. And it will return you a matrix, like a data frame with the read counts. So if I demonstrate this, the package is pretty fast. So you can see in the progress bar that it can extract like 100 samples in, like a fraction of a second. And then in this case, so if we go through, we can extract samples by index. We can extract samples by, again, the sample IDs. We can extract all the samples of a gene expression series. And we can extract all the samples that match a search term. And so these can be fairly large. But so in this case, we can see we found 1,000 samples that matched myoblast. And we removed all the samples that were potentially single cell data. And so you can see you can really, like, you're pretty agile with this tool to get the data that you might be interested in. So one thing to note, like this search is fairly primitive, I would say. So it's using a regular expression. So you can just type any regular expression in here and it will try to find it in the metadata, given the metadata fields that you provide. And it has some default metadata fields that it looks in. But you can add them. And we have a GitHub where it's like described in more detail. But so this is, as I want to like mention as caution, because the geo metadata is a little bit wonky. This search you have to be a bit careful how you search the metadata. And okay, so then the last group, which isn't like there's a utils package, but so there's a group of convenience functions that are really useful on a regular if you work with gene expression. And that's one is to deduplicate the genes, and then there is a function to filter genes based on their expression. So there's a couple of ways you can filter them, but in this example, we require that the gene is expressed, like has a gene count of 50 or higher, and at least 2% of the samples that we find. So you can, filter genes that way. And so this function also automatically can aggregate the gene symbols, in which case it will just sum up all the counts. And then lastly, we have this normalization. And the normalization has a couple of options. You can do a log quantile normalization, CPM, or trimmed mean to get a corrected count based on the library size. So if one sample had 2 million reads and the other one has 1 million reads, this kind of takes into account. And so if we run this, so these functions are pretty fast unless you're dealing with like a very large data set. So just to illustrate, so in this example, how we did it, we started off with 67,000 genes and 460 samples. And the samples matched one gene expression series in Geo. And then after we aggregate the gene symbols, we're down to 62,000 genes. And then if we do the aggregation and then filter genes out that have very low expression, then we end up with about 16,000 genes. But again, because you get these, I guess I can show it real quick. So the data frame looks like this, right? So you have the samples as columns and the genes as rows. And then after the normalization that we just did, we can see that, hopefully they're like somewhat normalized. So here you can see this is before normalization, and then after normalization, all the distributions are comparable to each other. In this case, we used quantile normalization, so technically it forces the distributions to be identical. All right, so that was the demo. And as I mentioned before, the R package will come out relatively soon, and then we'll put a link on the landing page of ARCHS4. And then this is the last thing I wanted to show. So this one I don't show live because it takes a bit of time to load, but this is the ARCHS4py alignment package. And so this is like a really convenient way of doing RNA-seq alignment because it has the ability to load the raw FASTQ files with like really easy commands. So you just specify the SRR IDs and then the output folder, and it will extract them from the SRA database. And then with the initial command that we have here on the top, we just tell it to align the FASTQ file that we just extracted against the human reference genome. And so what you can immediately see is that there is like a couple of steps missing that you usually have to deal with. So this package automatically downloads all the required reference genomes, and it builds the index if it doesn't already exist, and then does the alignment automatically. So instead of having to deal with all of this, it's just a 2-line or like a single line piece of code to do the alignment. And then in the second case, this would be for paired data. So in this case, instead of giving it just a single FASTQ, you give it a list of 2, and then it will do paired alignment. And then the other one that I like a lot is this alignment of a whole folder. So here, if you have a folder with a lot of sequencing data, you can just tell the ARCHS4py package to download them all at once, and it will return you a data frame with the gene counts that are identical to the gene counts that you would get from ARCHS4, how we processed it. And so this is also fairly clever because it can identify if samples are paired or not. And it's not even based on the file name. It will like look into the files and it will identify matching the matching reads by the read IDs. So it's like a very convenient way of just aligning a whole bunch of samples. Okay, so yeah, that was basically it. So I hope after this, if the server is still running, I'll see after this talk. You can try the functions for yourself. And so we just released the website. So if you encounter any problems, please let us know, and then we'll work on fixing those. So what I was talking about, just as a brief summary, we talked about the new ARCHS4 website that was completely redone. And we really hope that we, added like something really useful that people will come back to. And I think, like one of the things that really was more like an evolution than like a rewrite was this automated pipeline that we're running now that's very robust to all the issues that you can encounter when you have to process millions of files and like move them over a network. And so this kind of accumulated into what we now, can confidently say that we have monthly data releases. So if you come every first, I think, Wednesday of the month, or like maybe Thursday, the files should be in the download section. And we'll see if that works out without any of us, like with me not having to interfere or like fixing anything, but I'm hopeful. And then the new website that replaces the old platform, I think we really enhanced with also using and leveraging the abilities of the large language models to, really like boost the descriptions of the genes, and potentially we go into also better understanding the metadata in geo. And then we have the tissue-specific gene correlations, which I think is a really nice feature. And this is really something also that we use in PrismX. We use these tissue-specific correlations, and we can really show that can really enhance machine learning algorithms. So I think this is really valuable. And then we have implemented fast gene expression signature search that allows you to find similar signatures relative to your input. So if you have some gene expression profile and you query this database, you might find similar samples this way. And then we have the marker gene identification, which you could also, of course, compute if you download the big file. These things are easy to compute on your own, like on your computer, if you use the ARCHS4 package, for example. And then what was really a challenge was the new download function in the Data Explorer, where you can just click on a button and download it. And so what is also new is the series download. So try it out if you're only interested in a single series. Just sign in on the top right, and then you can download those zip files. And as we were showing, the Python package and R package, which is coming out soon, hopefully really useful for you guys. And I hope this was informative. So just thank Avi, who is the PI and of the lab, and that makes everything possible. And then all the lab members, because it's obviously like a group effort. Thank you for listening.